So now you should be able to access the practice questions here. I'll keep building on these, probably some variations on the existing questions. What I usually do is come up with a question that's trying to get at a certain kind of knowledge and write some variation on that so that you're not all getting the same question. You know, in case you're all in a room together taking the exam. I ask students and they often don't even know like 2 other people in the class, which is too bad. Probably not as much this semester. Not too easy to get to know people. This is also going to be my last in-person lecture for my 4 in-person students today. So Thursday we'll do what we normally do for the exams. It'll be available online during the whole of the calendar day for Thursday. And then I will be in the WebEx room. If you have questions, you can pop in there and ask them. Same format as before, so similar number of questions, similar types of like proportion of automatically graded and a couple of more free answer questions. And if you have questions during the 2 hours that you're allowed to take the exam, you can try to contact me. I will be available during the lecture scheduled time, and I might be available by email outside of those times. All of the usual stuff. People have done pretty well. This might be one of the few times, you know, this semester you get a chance to really see the exam, and I think the grades are often a reflection of how much time you've had to prepare for them. So I guess if you had infinite time and you could always look at the exam practice questions, eventually you'd get to pretty much 100%. That's the hope anyway. So I'm hoping you also reinforce your knowledge along the way. Don't just memorize correct answers. I think you know, if I've written the questions well, that'll be something you're required to do. What was I going to say? I don't remember. Any questions before we get going? I'm going to stop putting up the polls because nobody ever puts anything on here. But why not? Just throw it up there just in case if anybody wants to type in a question. Okay, so Thursday will be the exam. All of next week, so Thursday of next week is Thanksgiving. Tuesday of next week, they've asked us to be remote starting at the end of this week. So Tuesday we'll be meeting at this time. I'll be in front of my computer. You can choose to be in front of your computer. If you're not in front of your computer, I will record the content, same as we do it here. Just it won't look much different if you're at home, except I will be more of a face on the screen instead of a person on a camera. So you might actually see my face. It's weird. I had students, like when they reach out to me and we all have our masks off because we're doing it by WebEx. It feels weird. I don't know. At the same time, it feels weird to have a mask on and it feels weird to have a mask off. Yeah.
I get that.
Right? Sometimes I like forget that I have it on. Or I feel like I don't have it on and I do. Or should. I don't know. I want normal life again. Okay. Too much rambling. Not enough rambling. Hey, a question. Yes, the test will be available. I don't know what hours people keep, but I just put it on there for midnight to midnight. So at the midnight, the night at the end of Wednesday, it will be available until midnight of Thursday. I see when people do the practice tests, it takes them like 20 minutes to do it. So you probably don't need a whole block of time to do it. But it will be available during the calendar day. If you have any reason that time won't work for you, let me know. I guess not really. I don't have any reason not to give it to you on a different day, except it's nice to have it all wrapped up with a bow. All right. So if you have questions about the exam materials, store them up and we'll get a chance to talk about those. But I do want to wrap up the mutation content. This is something you've probably had before, as with a lot of the content we cover in this class. And again, I'm going to try to emphasize a little more the result. So we talked about how genes connect to phenotype. And in introductory biology, you probably learned your definitions. Okay, I know what a missense mutation is. I know how this can giving me a different amino acid sequence. And it's not a biochemistry class. We don't get into the function of proteins, but I do want you to think a little bit more about what could be going on with protein function. And function of protein would be connected to phenotype. So as we get started on our review-ish today, what is a mutation? Strict definition of a mutation. Noah.
Isn't it just a change to like the DNA in
That's,
a sort of way?
Yep, that's it. Real simple. So mutation technically is just a change to the DNA sequence. And the topic of mutation, we don't focus really on what causes the mutation, but we can think about what might be the effects of the mutation. And the effects of a mutation can be all across this big spectrum, right? So we can have a mutation that has a big effect. So let's say this disables an essential protein. And you can have a single mutation if it's in the right spot. This is the thermal vent on the Death Star, right? This is the back door breaking down something that was really important and might have been easy to break. It's possible. So if you did your yeast lab and we zapped those yeast cells with ultraviolet light, it's possible that one of those UV rays hit just the right spot, and that is possible. So I put here lethal question mark. There are considerations as far as whether that's a diploid individual and so on. But it would be possible under some scenario to do a major effect with just a single mutation, all the way down to small effects or really no effects. And there are a lot of ways for that to happen too. So what is an example of a mutation that might not really have an effect on the organism?
Wouldn't like a silent mutation be one of those?
All right, a silent mutation is a good example.
So like just a base pair, like changing, switching.
Whoever's talking is really quiet right now.
Oh, sorry.
Sorry. I couldn't hear what you said. You could try again, or? All right. What is a silent mutation? Noah.
I'm guessing one of the small ones that doesn't have any effect. Not one that you can see.
All right. Sort of. So it's kind of circular. Silent mutation is one of the possible mutations that would be small effect or no effect. It's not the only kind of mutation. So not every small effect mutation would be considered a silent mutation. I guess by the definition. So we're introducing the idea of silent mutation in a protein coding region. Jenna?
So that would be like a change in a single nucleotide that it codes for the same amino acid.
Okay, right. So a silent mutation, we'll stick with the definition of this here. So a silent mutation causes, so this would be a change in the coding region, the region of nucleotides that actually codes for amino acids, different codon, but it codes for the same amino acid. Okay, so it's silent because it is a change in the DNA. It counts as a mutation, but if you look at the sequence of amino acids that make up a protein, you wouldn't see anything different about those. Suppose if let's say you were baking cookies and you bought a different brand of butter. It's all still butter. So there's a difference, but the flavor of the cookies, at least as far as I can tell, would be the same if you used a different brand of butter. Different codon, same amino acid. Okay, so we explored the effects of single nucleotide changes. And that's a reasonable proportion of the mutations you might encounter. So a single nucleotide change can result from when DNA copies itself. Suggested previously or last time that there's a lot of copying going on, and it's almost inevitable that some mistakes are going to happen. So a single error in a single nucleotide, that's not too uncommon. There also could be environmentally caused single nucleotide changes like ultraviolet light damage. So of those, we saw silent mutations. We saw some that change a single amino acid. What was the name for one of those? Jenna.
That was a nonsense or missense.
Nonsense and missense are different things.
Oh.
So missense. So missense mutation is where a different amino acid gets put in. And then a nonsense mutation, a little bit different. The non part, non you could sort of think of as like nothing. So a nonsense mutation causes nothing to get put there. How do you put nothing into an amino acid sequence?
Stop codon.
Stop codon, right. So in a nonsense mutation, there can be a mutation that changes a codon from coding for an amino acid into a stop codon. So we saw some examples of protein sequences, polypeptide sequences. I might have done it just like this before. So there's a lot of variety as far as the effects that can happen to this polypeptide sequence. The missense mutation, if it's just one mutation that causes one amino acid to change, that could be a minor effect, could be bigger depending on the function of that amino acid. The nonsense mutation causes the amino acid sequence to stop, causes this polypeptide to be shorter. Basically, if that happens earlier in the sequence of codons, that would make for a really short protein, probably one that doesn't function at all. if it happens toward the end, it might just get away with being okay. If that last few nucleotide or last few amino acids were not terribly important to the function of the protein. And everything in between. So practically non-existent protein all the way up to one that's just a little bit different. So we kind of talk about these theoretically, but there would be, say in humans, there might be disease phenotypes that go along with different kinds of mutations. So if you have a protein that's important for cellular function and it's a little bit broken, you might not see a phenotype. If it's medium broken, you might see a mild phenotype. If it's really broken, you might see a severe phenotype. And so you have basically a genetic basis for the phenotype. All right, good so far? A lot of review. So these were examples of single nucleotide changes. Another type of mutation is insertions or deletions, and we had just gotten started with that when we ran out of time before. And I've been not showing you slides. Let's show some slides. So the insertion or deletion is the kind of mutation, and frame shift is how we describe really the result, the effect on the coding sequence. So I showed you an example last time of how adding one letter or removing one letter from this sequence could have a pretty big effect on the sequence following that. We could maybe use one of these examples here. We could look up the codons and see what we did to the old sequence there. Let's do that. Somehow. All right, let's see. I'll just type these in here. G, 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 A, G, T, G, T, A, G, A, T, C, G, T. We'll skip the base substitution. Yeah, we can do that one too. G, G, A, G, T, G, C, A, G, A, T, C, G, T. Base insertion. G, 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 A, G, T, G, T, A, G, A, T, C, G, T. A, G, T. Okay. Whew. So in the slide from I got this image from the textbook. They have this convenient spacer in between the codons. In the actual nucleotide sequence, there wouldn't be any indication of where the one codon ends or one codon begins. So that relates to the reading frame. Anybody want to go over reading frame? Or good with that one? So reading frame, basically the position of the ribosome determines which 3 letters count as a codon, and that just starts at the start codon and continues in blocks of 3 through the whole sequence. So the ribosome doesn't know where a codon is supposed to be. There is no supposed to be. It's just wherever it happens to line up. So if letters get added or deleted from the sequence, then the whole frame of those 3 letters shifts. So we can explore the effects on the amino acid sequence. Okay. I guess another thing here, we're just going to, I didn't see it, if it was spelled out in the illustration. We're going to treat these like they're 5' at the left, 3' on the right, coding strand of DNA, and we can figure out what the RNA sequence would be resulting from that, right? And not too tough to make a leap from our T's to U's. So this would be the sequence of the DNA, and then over in the table would be the corresponding RNA sequence. And that works as long as these are the coding strand. All right, so GGG, glycine. I'm going to go ahead and put some separators in here. AGT, serine, AGU, GTA, GUA, valine, GAU, aspartate, and CGU, arginine. Okay. Second one, that example was a single nucleotide change, not an insertion or a deletion. And I'm going to maybe avoid repeating myself here. In this case, we've got the GUA codon is now a GCA codon. GCA valine valine becomes alanine. Apart from that, the other nucleotide or the other amino acids would be the same. So glycine here, still the same codon, serine the same codon, aspartate the same codon, arginine same codon. So that one would count as a missense mutation. All right, let's see what else we broke here. So same insertion. Okay, so instead of GTA, we've now got a GTT or GUU, still a valine, so no problem yet. But wait, AGA, arginine, and UCG, serine. So one happened to be the same valine. This one's different, that one's different. Here we've got one deleted, GAG is glutamate, 8UC, isoleucine, and I guess we don't know what happened there. So schematically, I guess if I zoom out a little bit farther, depending on where the mutation is, things are basically messed up from that point forward. So practically random. So following an insertion or deletion, the amino acids change. And if they're different amino acids, you're building a different piece of protein there. So depending on whether you started early on or late in the protein, if you start early in the protein and you've changed most of the amino acids in that protein, it's not going to function in that way. Probably won't have any function, just sort of a lumpy piece of protein that's not going to do anything for the cell except waste time, take up space. If you have a mutation later on in the sequence, a frameshift mutation, that might change a couple amino acids toward the end, might not affect the protein. One more time, it depends. And also, really, by virtue of this basically randomizing the codons, 3 out of the 64 possible codons are stop codons. It's possible that those amino acids, one of those positions might turn into a stop codon. It's possible that an existing stop codon could get turned into a coding codon for an amino acid. But I guess it doesn't really matter, right? Everything after that mutation is probably messed up, whether it's amino acids that are the wrong amino acids or missing amino acids. Big effect. I guess some people, their whole career is probably mutating proteins and finding out what happens. We didn't talk about this one, and it's interesting to me. I frequently have a student raise this question, and as far as I know, there's not a name for it. So we have nonsense mutation, turning an amino acid codon into a stop codon. What if you had a stop codon and it turned into an amino acid coding codon? Possible. So this is my example of just can't stop. Drive off a cliff. Just kind of a random little bit of trivia here. This is from a study I found, and students raise good questions, and sometimes I have to look into the answers. And this is from yeast, and it turns out that they have like backup stop codons. A lot of them. Even at a specific position. I don't know if I could figure that out myself. So if the existing sort of normal stop codon is mutated and doesn't function as a stop codon, there's another one afterwards.
So what if the start codon was mutated? Would it just never get translated?
That's a new student question that I now have to put a slide in for. The question is if the start codon got mutated. Right, I was thinking about that myself.
Sorry.
I'll give you credit on the slide. Yeah, if you had your mRNA with a start codon or maybe there's like a if you mutated that start codon, the start codon is in an important position. It's basically the first AUG that the ribosome would encounter. And if it's not there, it's probably unlikely there's a second AUG also in the same reading frame lying near the beginning of this protein. So I'd say, just offhand, I think you're probably in a lot of trouble if you mutate your start codon. As far as I know, there's not, I can look into this one now, but I don't think there are backup start codons. And looking at mutations, we don't talk about evolution or selection or fitness in this class a whole lot, but you probably covered that in your early introductory biology classes. Mutations happen in cells. If that cell is part of a multicellular organism and it develops a mutation that prevents it from making an important protein, this is something that gets selected against. So it might not, just bring an organism crashing to the ground to have one cell with a mutated gene and certainly not being able to make important proteins. This is something that selection can take care of. That would cause an organism that, say, it has that widespread or throughout its whole genome, being unable to make an important protein. That decreases fitness, causes that individual not to survive or not to reproduce, and these variants are lost from the population. So I guess sort of keep track of that in the back of your mind. Organisms don't just accumulate mutation without consequence and then see what happens in the next generation. Good question. All right, here's a question for you. So let's say we had amino acid sequences like this, and the one on top, the gene that codes for it was mutated. What kind of mutation seems to have caused this? I probably won't put up the multiple choice, but somebody can just chime in if they want. So we have the same methionine, tyrosine, threonine, same, and then suddenly different. Isoleucine becomes tryptophan, glycine becomes serine, and so on. All these are different moving forward, and then it even stops early. What do we suppose happened there?
Frame shift.
Probably a frame shift, right? So I guess if you have any case where there's a single mutation and the result is affecting even more than one amino acid, then you're probably looking at a frame shift. And hopefully this maybe illustrates what I've been talking about. This basically kind of randomizes things after a point. So randomized these amino acids into different amino acids and then led to an earlier stop for the polypeptide. Okay. So I guess when I think about mutations and exam questions, I probably will have you working forward and backwards about this. So change the mutation in the DNA, what could be the effect on the protein? Or look at the protein and ask what could have been the mutation that caused it. All right. Any questions on that stuff? All right. Another thing you probably didn't talk about too much in introductory biology, kinds of mutations that might still affect the phenotype but are not related to protein coding. Or I guess not immediately involved in protein coding. So as we've seen, there's a whole lot of DNA that is not itself protein coding. What are some important DNA sequences that don't contain codons? I was putting exam questions together and it occurred to me I might dwell over this too much.
What was the
So a piece of DNA that you could mutate to have an effect, but is not involved in coding for amino acids.
That'd be like tRNA and rRNA.
Okay, that's a good one. Kind of a loophole to my question there. Yeah, so mutations that don't affect protein sequence. Right, so there are, as we've seen, genes for tRNAs. And rRNA, right, so maybe take a tRNA example. The tRNA has this three-dimensional structure. It's kind of a cloverleaf structure where some pieces of this are complementary across different parts of the strand. They make these so-called stem regions. And a mutation in here, in this region, might cause the two pieces of this stem not to be so fond of each other, and mess up that complementary base pairing. Certainly a mutation that affects the anticodon would be a problem. So we're not going to talk about the specifics, but I think it's nice to think outside the box of mutations. That would have a pretty big effect if you disabled the function of a tRNA. So tRNA, as we saw a little bit before, there's DNA. There's a DNA gene for a tRNA, and tRNAs are made by transcribing that. So a mutation in the DNA could give you the wrong configuration for a tRNA. What's another piece of DNA that might be important?
Introns?
Introns, right. So introns, I'm going to say intron splice sites or signals. Right, so we saw there are pieces of DNA that get transcribed on the RNA, and then they basically serve as instructions for where the introns are and that they should be taken out. We had the promoter region. So really everywhere we've seen that you can have a nucleotide signal, mutate that, then you run the risk of that DNA not functioning in the same way. And particularly for a promoter, that could be a problem, right? So the promoter is responsible for getting transcription to happen in the first place. If it doesn't work the same, then you might not get any transcription. It doesn't matter how good your amino acid sequence code is. If that never becomes an RNA, you never get protein. I guess a little more generally, they're using the term regulatory site. Probably part of the promoter, I would guess there.
Professor, your mic is, I don't know, there's an echo for...
Okay. Just noticed that my... Better now? Better now.
Now it's better.
Cool. I had never put the charger in the receiving box. I'm going to miss all the technology. I feel like a bionic person up here. When I teach class from my office, it's just me and a computer again. Not me and 3 wires and 4 microphones. Okay, so regulatory sites. We talked a little bit about consensus sequences. So these are some examples of showing the variation, possible variation at these intron splice sites. And this kind of study kind of highlights where changes are allowed and where changes are maybe less allowed. So if you look at over here, the 3' splice site examples, they all end in this AG. And the fact that those are really tall, that's like, they're vitally important. So those 2 nucleotides being A followed by G, that's really important. The next letters after those may be a little less important. So it kind of gives you a little window into what could be mutated and still allow this to function. So we sort of draw a conclusion backwards based on where we see variation, where we don't see variation in natural organisms. And not seeing any variation kind of makes the case that that really can't change and still be functional. So this AG at the 3' end, this GT at the 5' end, really, really important. Found in just about every intron of this type. All right, so mutations, back to the whole concept of mutation. Mutations can be detected by the cell. There's a so-called proofreading function of the DNA polymerase. So as it's making a new copy of a new strand of DNA, it is able to sort of check its work. So it puts one in there initially, and then before it's done, it says, was that the right thing to do? What's shown here, this is a bigger scale error checking after the fact. So after this new DNA strand has been made, there's one labeled new DNA and one called old DNA during the process of copying DNA. And there's a mismatch. So G mismatches with T. After it's made, how do you know who was the original? Good question. Right, so older DNA doesn't look any different from newer DNA, or does it? So there's a process where older DNA accumulates these methyl groups as it goes along. It's a little modification, chemical modification to the nucleotides. And having been around for longer, the older strand will get more of these methylated nucleotides. And that's a little signal for who's older. So when you have to tell somebody to kill their clone, or you got 2 identical, a person and her robot imposter, who's the real one? Why the movie's old by now. Terminator 2, in case you're curious. So the true, what's her name? Sarah Connor. The true Sarah Connor has the methylation because she's been around for a while. And the newly made robot Sarah Connor, no methylation. Okay, so this is one way that it'd be possible to detect that there's a mismatch. It does really kind of make this sort of chemical bubble of nucleotides not quite matching with each other. There are enzymes that can go through and check for that. However, it's possible that mutations go undetected, and there's a point of no return, basically. So at the right here, think about our 2 strands of DNA. As those strands get copied, the strand with the mistake in it would be this one that's got the AGGC instead of AAGC. It's a mismatch here, but after one more round of replication, the correct strand and the mistaken strand both get copied. And at that point, this is a double-stranded strand that has a proper nucleotide base pairing, just with a, we'll say, wrong or mutated or different sequence. So if the mutations aren't caught in basically one round of cell division, they will not be caught in the cells that result from that. And I guess back to the topic of evolution, sometimes this is okay. Change is good, variation is good. Sometimes. Mostly it's problematic to mutate a gene. Sometimes you get something good out of it. Another thing to think about is that, again, within a multicellular organism, there are a lot of cells that don't have any part in producing the next generation genetically. Pretty good, pretty clever job there, multicellular animals, organisms. So I can go through my life, I can get damage to my lungs and scars on my skin, broken bones, and I suppose genetic versions of that, so mutations to different cells in my body. As long as those are not mutations in the cells that go on to become my children, they're not going to get passed on. So there's this term, germline. I always find that seems a little outdated. Germline, like germinate, or germ is like the beginning of something. So your germline cells are your cells that are going to become sperm and egg cells, and everything else counts as your somatic cells. Those are your body cells. And sort of a nice protection against passing on mutations. Also, if you do have a mutation in one of those germline cells, then that can have a really big effect. So that potentially passes on the mutant version of DNA to all individuals that are inheriting that. All right, so I got maybe too much left. So I'll zip through a little bit faster. The end of the mutation slides are just some material to think about, really. So not every cell in a multicellular organism goes on to contribute to the next generation. Not every gene stands alone. So if you get a mutated version of a gene, most likely the first thing, the immediate effect of mutating a gene would be to produce an allele. And that allele would be subject to the rules that we've seen in Mendelian genetics, dominance, recessive. So don't worry too much about the specifics of these examples, but I just want to introduce the idea that some mutations can be dominant, some mutations can be recessive, and it comes down to the function of the protein and the nature of the mutation. What have we changed as a result of mutation? So probably the easier one to think about is what we call a loss of function mutation. So the protein is basically broken and can't do its normal job. So here's an example, rhodopsin, a protein related to color vision. And we've seen that color blindness can be due to a recessive allele, and that's a little more problematic in humans because it's inherited on the X chromosome, and males get one copy of their X chromosome, so a little bit greater chance of seeing this recessive allele show up in the phenotype. So there's a number buried in there. Okay, so probably the most common, easiest to think about, recessive loss of function. It's also possible to mutate and get a gain of function. I suggested earlier it's not very likely that a mutation is going to generate something positive. It happens. It happens on evolutionary timescales, thousands of generations, thousands of individuals. But for the most part, it's pretty unlikely that a helpful new mutation is just going to pop up. So that's where our comic books diverge from reality a little bit. So if you expose yourself to gamma rays, maybe if there were a billion of you, one of you would come away with something positive, but mostly you'd probably be messing up the good parts that work. But we can look back through evolutionary time and reconstruct what has happened. There are genes that are related to each other. So they basically have very similar sequences, but different functions, and they exist as multiple copies. This is an example still from a eye pigment, or let's see, an eye color perceiving protein. Our color perceiving opsin genes are related to each other. So they're similar. They were not built from scratch. They were built from a common early version of an opsin protein that diverged and became one that absorbs reddish colors and one that absorbed bluish colors. And this difference comes about through mutations from the ancestral original opsin gene, whatever color that saw. Becomes two different versions. So organisms that are different from each other, adaptive changes or random changes in organisms relative to their ancestors are the result of mutations. All right. So possible also to have, let's see, we've got dominant gain of function. This is more like a positive effect. This is a gain of function that has more of a negative effect. This is where a mutated version of the protein kind of gets in the way. So again, don't worry about the specifics, but this is an example I found where a single amino acid substitution causes this cell proliferation that is problematic. So this mutated version doesn't just get out of the way, it actually kind of gets in the way, causes trouble. So dominant gain of function, dominant loss of function. I guess that's maybe a little bit like gain of function. Another example, mutant protein. Causes a phenotype there. So lots of examples. We'll kind of skip over these. A little hairy for now. You're probably not ready for that today. So I did promise some time. I'm going to stop there and let you ask some questions. If you've had a chance to look at the practice questions, or if you just happen to have some questions from whatever we've gone over. So our content for exam 3 is molecular genetics, viruses, and central dogma, basically, all the way through mutation. Fire away.
You're making a protein from an mRNA strand. It's just like you just read the 3 and look at that chart, correct? Or the
Yeah, so if you are translating an mRNA into an amino acid sequence, you'll take them 3 letters at a time, and it's important to know where to start, though. So, nope, not enough. All right, so here's an example. Let me see if I can blow this up a little bit. Mine has the answers on it. Preview. Okay, so this one, suppose the sequence for a mature mRNA molecule is that one, 5 prime, and then this sequence, and then 3 prime. As I mentioned a few times, I end up having to shorten these just so we can basically think about them without going crazy. If this was strictly the mRNA molecule, you wouldn't start with the first 3 letters, right? People are often very tempted to do that, but you've got to think like a ribosome. So the ribosome attaches to the molecule, and it basically slides down toward the 3' direction until it finds a start codon. So you want to do the same and scan down here until you find the start codon. And there's one pretty close to the end there, the 5' end, so AUG. So you take that 3 letters, AUG, gives you methionine, followed by GCA, alanine, and so on. So it is 3, then 3, then 3, but very importantly, starting at the start codon. And then I guess you can kind of check yourself on these. I think in my examples, I'm always going to put a stop codon. So if you're putting your amino acid sequence together and you don't run across a stop codon before you get to the end, you might want to double check yourself. So I have one, this one is a fill in the blank. It's automatically graded as long as you follow the guides here about typing it in. So this would be methionine, abbreviation just as it is there, then dash. Then what do we have? AUG, GCA, alanine, and so on. GCA, AGA, GAC, CAA, UAA. All right, so I recognize the UAA because I put it there. And that's a stop codon. So there is a stop codon, about 5 amino acids or so down the sequence in the reading frame, and that would tell you to stop putting on new amino acids.
Do we have to write stop or anything just for like formatting purposes, or?
On this one, I allowed a variety of answers, so you could do not. So stop technically doesn't add anything to the amino acid sequence. You could just stop at the last amino acid before that. If you want to write stop, I understand that people might be looking at the table and typing the word stop in because they see it in the table. That's fine. Yeah, so as before, if you're going through the practice exams and something looks like it's the wrong answer for my fault, then please let me know about that. I'll just say it's bound to happen. There's a lot of questions, and I write variations of the questions, and this is my first time around with most of these questions, so if I make a mistake, usually it's like a multiple choice. I forgot to mark the right answer. Please let me know. I had on the last exams some people who were like training themselves on the incorrect answers because they knew the computer would give them credit for those, and I don't want that to happen because then you end up learning the wrong answers. Maybe you know the right answer, or maybe you just think that. Anyway, let me know about those. There's a help yourself point in it for you.
Will you be able to grade, like, if we get like a fill in the blank or something wrong, and then, but like because of the, like this one that you were just showing, and it had stop, but it said we counted it wrong because we had one extra part. Will you be able to like regrade that and then give us the points?
Yes, I can regrade things. When you lose points, I want you to like fully lose points, and I don't want you to lose points just because you're almost right or there's some minor detail. Canvas is very, I don't remember if I disabled the uppercase. Sometimes it's very particular about uppercase and lowercase letters. So immediately I'll get a grade for you that is the one that's automatically graded. I can change the grades if it's something really minor that you basically got the right answer but didn't get automatic credit for it. Noah.
Will you have any questions with like a more bacterial layout where you've got multiple starts and stops throughout the DNA strand?
Question about multiple starts and stops. If we had a bacterial gene, I don't think I put one in just yet. I have some like this. This one's a eukaryotic gene example. I have one that's also a bacterial example. On that example, I didn't put in multiple sites, but wouldn't hurt to be able to think about that.
Okay.
Yeah, so a lot of this, like this question here, again, I'm emphasizing a lot these signals. So you've learned in previous classes what happens, but I want to maybe reinforce why it happens. So there are enzymes responsible, and the enzymes detect part of the sequence. So they don't just know everything and they know to come in, oh, this is the place exactly where I need to start chopping out an intron. There's a piece of the DNA that becomes the RNA that basically tells the enzyme where to chop. And that's what I emphasize a little bit more in this class.
There was a question about like anticodons on there. I don't remember what it was, but I was kind of confused. Can we look at that one?
Sure.
Yeah, I think it was that.
Yeah, the old anticodon thing, that's what usually I'm asking for is something that people might not be thinking about. So if you have a piece of mRNA with a codon, the codons in the codon table are written 5' to 3' left to right. And then I guess I'll put it down at the bottom here. The tRNA interaction with the mRNA is anti-parallel, just like all the other nucleotide interactions. So if this is running on the top strand here, 5' to 3', and the bottom strand is running in the opposite direction. So it'd have 5' over here and 3' over here. So I have a few questions that are basically variations on making sure that you know that nucleotide base pairing is anti-parallel. So knowing that you probably are okay getting the letters that are complementary to each other, but just keep track every time the direction of those letters. So I guess we can fully answer this question that's up there. So one possibility here, as I've written it, is five prime. Now we're looking at GCA. I think I already need to change the answer to this one. Thanks for letting me know. All right. So let's look at this second part here, wobble pairing. What about wobble pairing? What wobble pairing more often does how much detail do I want to go in here? One of the things we saw with wobble pairing is that the same tRNA can interact with, say, two different codons. So if we did have UGC. Oh my goodness, I'm sorry. I sympathize if this is challenging, because I am getting it wrong too. All right, let's get our arrows in the right direction. ACG, anticodon, we're going to go 3'. 5' is complementary to UGC, running that direction. The same anticodon could be complementary to UGU. But that is not the way I asked the question. I was asking about different anticodons that could pair with UGC. And it's this third nucleotide position that participates in the wobble pairing. So the way I asked the question, C is not a nucleotide that participates in the wobble pairing. So there should only be one anticodon that interacts with that. And I think I have to change my answer key on that one if I remember right.
So it's just G and U that can do the wobble pairing?
Yep, just G and U, and for codons, the third position of the codon.
Okay.
So I'll probably add in some variations on this. I'll maybe flip codon and anticodon and ask the same question backwards.
It's the third position on the codon, so would that then be like the 5' end of the anticodon?
Right. Yes.
Okay.
That would be the 5' end of the anticodon. Right. So this this nucleotide here is the 5' nucleotide out of those 3 nucleotides that make up the anticodon. So it's a G, but it could bind with C or wobble with U. Sorry, that one's probably proportionally a lot more hassle than the small concept is worth. Other questions? So I played a little bit with some of the functionalities. I've got put them in order. I've got some maybe one. I don't think I had any click on the image. Maybe I did. Matching, so letters or numbers standing in for different kinds of recognition sites here, Intron, Splice site, start codon, and so on. Mostly multiple choice. So I think right now I have 20 kinds of questions. So it'll be roughly the same as previous exams, about 20 questions automatically graded and then 2 free response. All right. Going once. Going twice. G, C, G, C, A. All right. If you're paying attention, this one will be fixed, as we described it. Anti-codon. G, C, A. Okay, so next time I'm live on Tuesday next week, I'll be in front of my computer. It'd be great if everybody wanted to turn on their cameras. I know that that's not always done. And I like seeing faces. Maybe you miss seeing faces. If we all do it, it won't be weird. If I'm the only one whose face we can see, then it'll be weird.
I'd have to go buy a webcam.
Yeah, the technology thing. We take it for granted. It's just in everybody's hands, but that might not be the case.
Yeah, just take a video of yourself and like.
Yeah, I'll do one of those background things with just me.
Yeah, I didn't think about that. So how will you just do it on a computer without a camera?
I'm gonna be on campus. I'll just come sit in here.
Yeah, right? I'll still be here, just not here. All right. I guess I will turn this off. Have a good one.
Thank you.
Thank you. Good luck on the exam.
I just had a question.
Yes.
So you said that capitalization matters during the exam? For like when you're doing when you're writing like the amino acid abbreviations?
It's so the way that this is graded, I'll show you what it looks like on my end. So I type the question and then I write in the correct answer, and then I have it basically where you can type the answer, and I have specified correct answers. The answers I specified, I think it holds you accountable for the capitalization there.
Okay, because I was gonna say like I had the right answer, but then it was telling me it was wrong. So then I realized it was because I didn't capitalize them.
Okay. I mean, I can add a whole bunch of possible answers here.
No, you don't. I mean, you don't have to do that. I just wanted to make sure so I know for like the exam.
You know, it's okay. When I fix them like this, it's one less problem I have to chase down later. But like I said, ultimately, we just if you got the question wrong for something simple like this, you can let me know and I can get you credit for it. But that would have to happen on the back end. Any other questions?
I think that was it for me. Thank you.
Thank you. Contains asp. Well, I guess if you're all set, you're the last one here. I'll sign off unless you got another question. Okay.